Good morning, everybody. Thanks for joining us today for a tech team meeting. We did not meet. Our last meeting was going to take place during the Great Salt Lake Issues Forum, so we all joined Lynn, and that was fantastic.
Thank you so much for that.
Yeah, it was a great forum. It was so good to be back together with everyone and see Lynn happy that we could have that event once again. So that was fantastic. Let's go ahead and start off with some introductions. We have a full room, and then I'll go ahead and go over the agenda. For those of you who can't see it, there are a few copies here if you'd like. So I'm going to send around also a clipboard if you wouldn't mind signing in. And go ahead, Elliot. Start you.
Elliot Chegnecki, Utah Geological Survey.
Andrew Rupke, Utah Geological Survey.
Mondo Masson, Central Data Sewer.
Lynn DeFreitas, Friends of Great Salt Lake.
Tim Hucks, Brancham Cooperative.
Bonnie Baxter, Great Salt Lake Institute, Westminster College.
Bill Johnson, University of Utah.
Wayne Wurtzball, Utah State University.
Oh, Jennifer Biggs, Forestry Garden State Lands.
Jamie Barnes, Forestry Garden State Lands. Ann Neville, the Nature Conservancy.
Don Hartman, U.S. Magnesium.
Carly Beadle, Westminster Great Salt Lake Institute.
Lake Bingham Water Rights.
Jim Van Leeuwen, Wildlife Resources.
Bernadine and Justin Miners.
All right, very good. Well, we have a lot of folks online. I'm just going to direct those in the room to the folks on, and you can see who's online.
Laura,
You got a whole row behind you.
Sorry. Please go ahead.
Todd Johnson, elephant in the room. I'm a professor at Utah State University in the Department of Landscape Architecture and Environmental Planning. And we are here under the kind of auspices of an initiative that's funded through a foundation called Land, Water, and Air. You've probably heard about it. The Quinney group has funded that universities and all of its departments to look at different facets of climate change and especially focusing on the basin. So we had a wonderful lecture by Wayne last week, and I have 11 students that are at the graduate level, and we are going to be shaping gathering some knowledge, putting together some thoughts based on kind of representing the Nature Conservancy, the Weaver Basin Water Conservancy, and you all, the advisory board, the tech team, vis-a-vis advisory board. So we're going to shape 3 initiatives that enlighten both them and all of us regarding those 3 stakeholder groups. So we're here to observe you and to hopefully make some contacts for information and involvement in our studio as we move through the semester and then into the spring with another initiative with our seniors. So that's
That's fantastic.
reasonably clear.
Corey Gertzman.
Joe Pantos, Utah.
Joelle Whitson. Amy Reed.
Andy Hughes.
Addison Martin. Amy Gunther. Emily Osen. Mickey Vigil.
All right, see a few people trying to trickle in. All right, so on the agenda this morning, we have Great Salt Lake Advisory Council updates. We have a Salinity Advisory Committee update, tech team member update. We'll go over the hot topics that were funded in FY23. Those projects now are well underway. And then Bonnie will give us a discussion on microbialites and primary production in Great Salt Lake. And then Pace VanDevender is on the line, and he's going to be talking to us about his research that he was doing on Quark nuggets over the Great Salt Lake. So we're looking forward to these presentations. I'll kick things off talking about Advisory Council and tech team updates. Jeff Denblecker is joining us in a few minutes, and he's going to talk a little bit about the Salinity Advisory Committee and some of the actions that have taken place as a result of that group's work. So if I could, and I guess if there's anyone out in the virtual world, if we can all, if anyone has any questions, I guess at any time, raise your hand and give us a holler and we'll have you join in the call. It looks like there's, how many people? Let's see. About 21 folks online, so welcome everyone online. Sorry, got to acknowledge that. If I may start off first, instead of talking about advisory council updates, I would like to talk about the hot topic grants that were funded in 2023, because part of that discussion will lead into the advisory council discussion. So I just wanted to let you all know which projects were funded by the tech team this past fiscal year or this fiscal year. And the first one is, these are actually not in a particular order, just the number they were on our spreadsheet. But so one of the projects is microplastic transport through Great Salt Lake through the Logan and Bear system. Oh goodness, sorry, my apologies. Not the correct list. All right, sorry, 2023 grants. We're continuing a study that UGS did last year, water delivery to wetlands. This was a wetland water budget and wetland change analysis for the Nature Conservancy's Great Salt Lake Shorelands Preserve. Number 2 was a dust study. It was assessing the vulnerability of northern Utah communities to dust from contaminated and growing Great Salt Lake playa. The third one is a USGS study that is the Causeway Breach. Is a Causeway Breach study looking at improved discharge and water velocity monitoring of the bidirectional flow at the Great Salt Lake Breach. Number 4 is Bonnie's project by Bonnie on microbialites measuring the resiliency of Great Salt Lake microbialite cyanobacteria. And let's see. And then the 6th one was we're continuing to work with Sam Duda on Causeway Breach flow. So the multi-fidelity modeling of flow and salt transport through the West Crack Breach or the Causeway Breach. And a couple of the projects that were lower down on the funding list, but the Great Salt Lake Advisory Council was interested in funding are a LiDAR mapping project. So basically a test plot for LiDAR mapping on the bathymetry of Great Salt Lake. So mapping the floor of Great Salt Lake using bathymetric LiDAR, implications for core and tree faults, microbialites, and resource management. That's a UGS project. And then finally a shorelands shorebird study through Sageland Collaborative. So at the last advisory council meeting, the advisory council voted to fund those 2 projects. So coming from the list of tech team projects, we have those 2 projects. Any questions on tech team grant funding for FY23? Okay, seeing none, I will move on to advisory council updates. The council, in addition to awarding or getting going on this year's projects, has been busy with, or the projects that they have authorized have been moving forward quite productively. They did a lot of project work related or focused on the Causeway Breach. This past year, they funded a salinity monitoring work by USGS, Causeway Breach modeling by USU, and then that led to some more Causeway modeling work. And I think a lot of that Causeway work was basically looking at if we fill the Union Pacific Causeway with rock, how that will impact salinity in the North and South Arms. So the Salinity Advisory Committee, which Jeff will talk to you a bit about in a bit, and maybe those on the Salinity Advisory Committee could actually talk about that since Jeff is a little late. But those groups, based on the funding that the Advisory Council was able to come up with, Sam was able to do his work and channeled the, or gave enough information to the Salinity Advisory Committee to make some proposals on modifications to the Causeway Breach. So that happened, as you all may know. One of the other exciting advisory council funded projects that I sent out to you all a while ago was the hydro mapper. And I hope you all had a chance to take a look at that, but it is a one-stop shop, a go-to place for all things Great Salt Lake, with salinity, lake level elevations, flows from the watershed, from the Bear, the Weaver, and the Jordan, and reservoir sites, snowpack, all sorts of stuff upstream. And we should say it is an evolving work in progress. And we hope to continue working with USGS on that. And Christine has been awesome in helping with continuing communications on that. So please let her know or let me know if you have questions or concerns. It's a great tool that I've used numerous times and I've referred people to numerous times. So super appreciative of that.
Can I ask about the brine shrimp data on there? It's still not represented. The brine shrimp heat map for the matrix.
Oh, the brine shrimp matrix?
Uh-huh.
I don't know that the timing of that was such that the creators were able to put that on there. Do you remember
I
any?
think that we published the hydromapper before, because you're talking about
Before
kind of
we finished the
salinity
matrix.
Yeah.
Yeah, I think that was slated for phase 2.
Yeah.
So we basically run out of funding and we need kind of another phase to include other things of interest, which we're more than willing to do, but we're kind
So
of.
brine shrimp's on there, it just has no data. So there is a band for them, but there's no color on the.
So that must have come from the elevation matrix and the comprehensive management plan. That's what Toby and Shannon were using to build the website, I think, but we can check.
Well, yeah, and
Okay.
And let me get with you on just what exactly we're wanting in there to illustrate. And then also, like, we can also start a phase 2, like, what do we want to see next? How far can we take this tool?
Yep.
That could be one of our next items, and we can just have a growing list and continue to work with USGS on getting those items on there.
Can I ask a quick question about that? So that's being funded solely through Forestry Fire State Lands, or is it also coming from USGS?
They're a match.
Yes,
A match.
match.
In the long-term prospect to keep it going, what does that look like?
I think everyone's really optimistic about it, and we happen to have a lot of matching funds at the moment. That's kind of unpredictable moving forward, but I know there's a lot of interest, and we know it's important to this group, so, we'd like to continue supporting it to the best we can.
That's great, because, we had a version of this a long time ago through Utah State, through Dave Tarberton and Jeff Forsberg, and that ran its course, and now this is active, which is great, but it would be great to make sure that
It keeps
this
going.
doesn't just fizzle out. Yeah.
Great. One other thing, a project that the council supported was the equation of state, or updating or refining the equation of state with USGS. And there were some holdups due to some COVID issues, but it sounds like USGS has gotten back the data that they need, and they're wrapping up their equation of state report soon. And so we'll be able to have that information. But I don't know, Christine, if you want to just jump to the punchline on that, or if you want to save that for another day.
Yeah, the punchline is it still works. The equation of state we developed in 2011 it applies today with data collected a decade later. So we're really excited about that. And in addition, we've been able to extend it to higher densities. There was kind of a limited range we were operating under before, but now we found that we found really reliable results with north arm water and with water moving from north to south in the breach. So we're optimistic we can extend it to higher densities, which is important as we enter kind of a more saline condition in the lake. So it worked out well, and we hope to publish that wrapped into another salt study, soon.
Thank you, Christine. Just moving forward with the advisory council, they're meeting tomorrow at Antelope Island. We're getting updates on the visitor center expansion. As you recall, during the legislative session last year, money was allocated to expanding the visitor center. So we're looking forward to seeing what will happen there. And then Jim Steenberg is going to do a presentation on lake effect snow and how that may be impacted by low lake levels. And then also we'll be talking about the causeway modification. which didn't make it on the agenda here today in great detail, but Bonnie, Bill, Wayne, what do you all think? Do you want to take the salinity discussion update and then?
Jeff's here.
Oh, I didn't see you come in. Come on up here. So I'll just transition. Oh, and I do have one last advisory council update item, and that is, and I'm not sure how many of you heard, but Dan Tuttle, an advisory council member from the beginning, passed away prior, or just after the last advisory council meeting. So super sad. They're looking for a replacement for him. The Senate just confirmed a round of council members. He was one of them to be confirmed, but his space is now not confirmed. So anyway, we will miss him and his enthusiasm for the lake. So with that, I'll turn it over to Jeff, and you can talk about salinity, what the salinity group's been doing, and anyone else chime in.
Yeah, sorry to hear that. Dan was a great guy. You will be missed. The Salinity Advisory Committee's been very busy. I know if any of you attended the Great Salt Lake Issues Forum, you heard quite a bit about the work that they've been doing over the last few years. I think that a lot of the focus over the summer has been what can we do with about the increasing salinity that we're seeing in the South Arm. And so really starting a year ago, we started talking about this possibility and what can we do about it. And the one trigger or one tool that we had at our disposal was to modify the berm at the new Union Pacific Railroad breach. And so we evaluated that. We had Dr. Sam Dutta at USU develop this model to look at some options. And in the end, we recommended that we raise the berm 4 feet with the goal of trying to reduce or stop the north to south flow where we're importing salt back into the South Arm. The goal is to try to maintain as much as possible the south to north flow and the connection between the two arms. And so that was completed this summer. I believe it was finished on July 28th. It took them only about a week to do. And from the USGS data, they had a great uplookers, ADCP sensor in there, and they could see definitively that the north to south arm flow stopped almost entirely, and while we also maintained the south to north flow. So in terms of our goal of trying to reduce the flow of or import of salt from the North Arm, we were able to accomplish that goal. And certainly that the monitoring that USGS and others are doing is continuing, along with the modeling that is being closely coordinated with the monitoring up at USU. So now we had another meeting in August to kind of review the progress of that, and certainly with unprecedented conditions, we have new questions that we need to answer. And so just trying to understand, well, what is happening now with the salt mass in the South Arm? And as USGS reported to us in August, and reviewed for us in August, that we still have this difficult question, why is the salt mass in the South Arm increasing? It's not just because of evaporation. We still see the salt mass increasing. And so trying to understand that question is certainly a priority. But also, I think some of the other questions that were posed is, well, let's start thinking about what else we can do. As the lake, if the lake continues to decline, what other options do we have? What are the potential impacts? But not only that, but also trying to think ahead and saying, well, what happens if the lake does come back up? And so I think that the great thing is, I think looking back on the research plan that that the committee developed 2 years ago in 2020. We were trying to develop the science to anticipate some of these questions. I think we did. And, but now I think as the committee moves forward, is really trying to look back at those same questions and saying, okay, now what do we need to be looking at? And trying to develop the science, the modeling that we need to be able to play through some of these scenarios of if the lake continues to drop, or what happens if the lake does come back up? What does the existing berm do now? So that I think is the role of the committee, is to try to anticipate these things, to advise the state in developing the science and the tools needed to be able to better manage the lake and help the state be able to make better informed decisions.
Can we talk really quickly about the salinity range and where we're at today?
Oh, yes. So last year, the committee developed a matrix, a salinity matrix. And the matrix really looked at all the different beneficial uses of the lake, and we contemplated, well, what happens to that use as the salinity fluctuates? And so as we looked at all of these different uses, it's a pretty extensive matrix. We identified a range of 120 to 160 grams per liter of salinity as really kind of the ideal range where we can maximize the benefits to the maximum number of beneficial uses in the lake. And this is for the South Arm. I believe in last month, the salinity was at 171 grams per liter. I'm not sure where we are right now, but the concern was that we're above that ideal range. And certainly the concerns with the rising salinity pertain to, we've had the microbialites who we feel like are starting to decline, or we anticipated would start to decline at salinities above 160. I think that was the first threshold. But then also looking at the brine flies and brine shrimp and concerns with what rising salinity will have on those populations, and then having a ripple effect throughout the ecology of the lake. And so those were, I think in our August meeting, also we heard from some of the mineral extractors, they're having to kind of anticipate and retool, not just because of the lower lake levels, but also with rising salinity in the South Arm. That means that, we're getting closer to saturation, and that affects their operations. And so they're having to anticipate that and retool what they're doing for that as well. So, rising salinity certainly is not unprecedented in the history of Great Salt Lake, but certainly in the recent times does have an impact on the uses. So those are some of the challenges that we're trying to address.
Thanks, Jeff. Jamie has a question.
Hey, I know that there's a bunch of people in the room that have been measuring salinity. Who has the information?
About 184.
Yikes.
We measured, 180 last week and took profile samples yesterday, so we'll get confirmation from that.
You're back in the lake.
Yeah, Jim took us out yesterday.
All right.
So
Great.
we'll have confirmation from the center of the lake, so as of this afternoon, hopefully.
Did you hear Jim, Jamie?
Yeah, thank you.
So the saturation is halite saturation that the extraction companies are specifically worried about?
Yeah.
Okay. And then just in terms of the berm placement, it's static, right? So it was the right move to make. It was a cheap way to get this in place, but it will clearly need adjustment because no matter how much simulation you do, the system, you can't anticipate where the system's going.
Right.
So there'll need to be adjustments made. How much did it cost to put it in place, and is it feasible to keep adjusting it on the fly?
It cost about $500,000, and we have money allocated now and into the future to make adjustments as needed.
Yeah.
So we'll be ready and available. And actually, it was working with the railroad and the Army Corps was a great experience during this process. They were super willing to work with us, and I know.
Well, my experience with the railroad is we paid the money for a permit and they never responded. So
They were
That's
so
great.
helpful. They and perhaps it's because they didn't want their causeway to be messed up by anyone else but them, but they ended up like, we used their rock, put it on their rail cars, take it out to the spot, they dumped it, and then it was just the contractor that placed it into the water.
Good to
So
hear.
it was, yeah, it was a great experience, and it would move super quick, so. I'm optimistic that if we needed to get in there, you know, at a short-term notice, it would be no problem. And then we have the funding to do that as well.
And part of the costs, part of the monies that were invested to build the berm included building a ramp down to the berm. And so that ramp was left in place. So that's a cost that we won't have to incur again. So I think with the funding that's available, I know, certainly the committee will be keeping a close eye on that, on what happens to the lake. And that's a scenario that I think is certainly planned for, is if the lake does continue to go down, we're going to have to modify the lower berm. Right.
Jim, with the 184 grams per liter salinity, how are the shrimp looking?
Well, right now they've grazed themselves out of food, so we got low oxygen. They're coming to the surface suffocating. So, but that's somewhat normal. They go through doing bus cycles. What we need right now is a little bit of more. We need more nutrients for algae blooms because they're out of food. So the saving grace right now for brine shrimp is microbialites in the periphery. It's got oxygen there, you got food availability there. We just need some rain, like we're getting today. Just throw in some nutrients and it should kick off an algal bloom.
Yay, yay for rain.
Yeah.
Okay, thank you, Jeff, and thanks to everyone on the Salinity Advisory Committee. It's been an interesting journey, and super happy that the state had this tool that we could make these modifications to try and help out the south arm of the lake. Who knew that we would have been needing to do this a decade ago? So anyway, if nothing else on this, when are we meeting again?
October 12th.
Next Salinity meeting. All right.
So, Bonnie, are you going to then talk about the fact that, what the optimal range for microbialites and where we are now? Okay.
Yeah, I will.
All right, before we get on to Bonnie's discussion, tech team members, do you guys, I know so much has been happening on the lake. Would anyone like to report projects, issues, problems that they're seeing given low lake levels, great opportunities they're seeing with low lake levels? Tech team members, please feel free to step in and let us know what you have going on.
Laura, I have an update for Phragmites.
Great.
So we finished aerial spraying for the year. Forestry Fire and State Lands sprayed about 4,400 acres of Phragmites, and that ranges from the South Shore all the way up to the refuge. DWR also sprayed all of their WMAs. Our biggest limiting factor this year was drought. Most of the Phragmites, we caught it in time, we sprayed early, but we had actually additional funding to spray more, but couldn't due to drought. And the good news there.
The Phragmites dries up too much and it's not receptive to the.
Yeah, so what we found is spraying dry Phragmites just doesn't work at all. So there's no point in putting the chemical out there.
So if we just dry things up more, we're in good shape on Phragmite.
Well, then you can't kill it.
On Phragmite.
And then, so dry Phragmites is the hardest Phragmites to kill. If it dries up, it just stays there, waits for water. It'll wait many years. Once it has water, it grows again. The good news is we have plenty of funding to follow up with all of that work, so everything will be mowed this winter. And then next spring, we will do more revegetation, which we are now incorporating into our treatments. And speaking of revegetation, we had our most successful seeding this spring, and we had some monsoon rains that saved it. But we really densely vegetated 15 pretty strategic acres in Farmington Bay with broadcast seeding and disking. So we're super excited to see that. This is like the first time we feel like we figured it out, and if we can repeat this, we've got a revegetation tool in the toolbox, which we've been missing. And so with that successful seeding, which was in collaboration with Karen Kettenring's lab, the Wetland Ecology Lab at USU, we hosted a tour last week to show everybody what we learned and what we were seeing, and we had a great discussion. We had really good turnout. And then the day prior to that, we planted over 3,000 plugs in Farmington Bay outside of the Teal Lake unit. And that is part of Karen Kettenring's 4-year EPA project, where it is a demonstration project. So we're doing both implementation and actually demonstrating restoration on the ground, but they're also studying it. So it's incorporated into a couple master's projects. So really cool project, and excited to see that keep moving forward. And thanks to everybody that came to the tour. We had a really good turnout.
What are you seeding with, a variety?
So this spring, the mix was majority alkali bulrush and inland saltgrass. And those are the two species that we found we are able to get to rapidly germinate. So we're doing very targeted revegetation where we're putting the seed on the ground. Hopefully it's all germinating within 5 days and rapidly claiming that ground. And then with the plugs, we have those two species as well as a handful of others. So we're trying different grass species, some different bulrush species, and just continuing to search for like what are our most successful species for claiming ground and preventing Phragmites reinvasion, and then also getting the habitat that we want. And so we're kind of we're looking for like what are our workhorse species that we can count on to claim ground, and then the next phase will be then how do we build in biodiversity and more targeted habitat improvement.
Thank you, Keith.
That's all.
Any other tech team members or anyone that has great like update? Oh, Bonnie's gonna go ahead, and then Brian, you'll be next.
So I just wanted to mention that one of the concerns that I've had out sampling microbialites are the flies. And I know there's not as much science done on the flies because they haven't, had this commercial interest over time. So what's going on with the flies is terribly concerning, and we know that their salinity range has now been exceeded. The pupa are dead essentially, and I've been talking to Dave Herbst, who is at UC Santa Barbara and UC Santa Cruz, and he is the world's expert on brine flies and hasn't done any work at Great Salt Lake. But I'm hosting him with Lynn and Marcel with National Audubon to come out and do some work on these flies. I have some flies in the freezer from 5 years ago that Kate Barrett, working with Gary Belowski, collected, and Gary's going to send me those flies. So we hope to do some biochemical analysis on the old flies compared to the new flies. But he'll be in town for a few days, and I'd really love to have some meetings with folks. We're going to be out the lake a couple days, but I'd like to have some meetings with folks who may be interested. So hit me up if you want to be on that list. Because
What are the dates?
It's October 25th and 6th. So just let me know if you're interested in being involved.
Thank you, Bonnie. Anyone else?
Yeah, just a quick note about, we have a project from the National Science Foundation to try to understand the pathways of elevated mercury in the biota. And so we're using isotopic signatures, we're collecting spiders, pine flies, going to the water in the muck as well, and shrimp, of course. And so probably worth talking. But I want people to know because maybe Great Salt Lake Ecosystem Project, we should be thinking about whether should be collaborating there. But we're working with the USGS Mercury Lab in Wisconsin, who are the experts on the isotopic signatures and bringing that capability here.
From when to when is your project? How many years?
It'll go over the next couple of years.
Keep us posted.
Okay.
Anyone else? Tech team or?
I think I might just add from our sampling yesterday, we did see a deep brine layer in Carrington Bay, but not in Gilbert Bay. So just a touch of one.
And what site's in Gilbert Bay?
AS2, which is kind of our deepest site that we usually sample, and we usually pick it up if it's there. But
And is it on the north end of the Gilbert Bay or?
it's kind of on the southern end of the.
Okay, like USGS is 3510 is down there.
Yeah, close to
So
there.
nothing there. Okay.
Oh, Brian.
Good morning, everyone.
Thank you for
Apologize for not being able to be with you this morning, but just from USU side with Sam Duda, I wanted to just give a very brief update if that's okay. So with our 2023 grant, one thing that we've done in collaboration with USGS is we've relocated some of our sensors that measure salinity, and we've now placed those directly in the breach at 2 different elevations. And so we're hoping that that new data set will give us some new insights into how the new berm height has influenced the exchange flows through the breach. So I just wanted to let everyone know that we've got some new sensors in new locations.
Fantastic. Thanks for the update, Brian. Anyone else?
Yeah, I just wanted to say USGS has been monitoring the breach after the berm modification to kind of verify our continuous data streams that are coming in. And as Jeff alluded to, we're not seeing, when we go out there, we're unable to measure north to south flow. The velocities are very low. But anecdotally, when there is a change in wind direction, we do see North Arm water spilling over that berm and kind of entering under the breach. And it's been about over a month, maybe a month and a half since the berm was modified. Our team was out there last week doing another measurement, and we're still seeing north to south arm kind of plume of water. It's just hanging out there. And we've been curious to see if it would erode away as south arm water moves to the north. But it's kind of hanging out at a fixed elevation, and it's not necessarily diluting, and it's not necessarily going away. It's not clear that it's moving, but it's there. So just wanted to check that.
Do you know how far below the berm that interface is when it's not windy?
We've consistently been seeing at 4183, so that would be like 6 to 7 feet below the water surface.
Below the water surface.
And I think the berm, if we look at it, it looks like it's about a foot under the water, maybe a foot and a half. So does that give you an idea?
Under the water or under the berm?
The berm is about a foot or foot and a half under the water, and
Okay.
then we're seeing the interface about maybe 6 to 7 feet below the water surface. So I guess that would be 5 or 4.5 feet below the berm.
Okay, so there's lots of room to.
So south of the berm?
Yep.
Okay.
It did, so.
But it sounds like there's a lot of room to lower that berm to let more salt come from the south to the north and replace with fresh water.
Well, that would allow more flow from north to south.
Oh, so do you think that, okay, so it's about optimal in terms of keeping that north to south flow from coming over, it sounds like, what you're saying.
It's just, well, it's not moving. Yeah, the water's not, we're not moving. We can't detect flow from north to south.
Yeah.
It is still there.
I just wonder if we have room to drop it further to allow more south and north to export salt and
Yeah,
bring it fresh.
perhaps.
There's not much head elevation to move water in the first place. It's only gonna-
True, at this point, yeah. I'm anticipating runoff, but who knows?
Brian, go ahead.
I would just add to that conversation that what Christine was talking about, that's one reason why we relocated our sensors. And definitely from the CFD modeling we've done, we think we have a good idea of what lowering the berm would do in terms of north to south flows. We would think that those would increase, not necessarily strictly increase the south to north flows.
Right. Thank you. Anyone else? Updates? I should throw out a plug. It's not too early to be thinking about a hot topic grant ideas for 2024. So this next tech team meeting is, I believe, December 13th. And so by then I'm really going to be, I was going to say pestering, but asking strongly for hot topic ideas by that meeting. So it's never too early to be thinking about hot topic ideas for 2024. So if you have any now, you can send them my way, but in December, it would be great to start a list going. With that, Bonnie, I'm gonna turn it over to you.
Okay, yeah. You seen that? Carly, do you want to switch slides for me? Let's get my copy. Just don't roll around. They get mad when you do that. So I thought I should present. So it's presenting? Looks
It's
like
all,
it is
it's
on
full
mine.
screen on here.
Yeah, weird. Okay, it doesn't look like it's in presenting mode, but I think it is.
Oh, wait. This one.
After 2 years, you think we'd be done.
I know.
There's two different things happening.
They're all different. No, but it's presenting on my computer. Here. Okay, we'll just walk it through. So this is work that I did in collaboration with Eric Boyd at Montana State. And Eric Boyd had two grad students, Eric Dunham and Libby Phones, and Joel Carpenter was one of my students at Westminster. Eric the elder, Eric the younger, and who we called Eric the third, Libby, came out and were known collectively by my lab as the Erics. So I will refer to them as such. So we were interested in trying to determine how much photosynthesis is done by the microbialite mats versus the water column and try to quantify that. And go ahead, next slide. No. So it's advancing there, but not here.
Yeah, I'm just gonna click through.
Okay, just click through them. Yeah, that's fine. So I'll just start out by saying the things that all of us in this room know, that we've got microalgae. Some people call it phytoplankton. I just got dissed in a paper for calling it phytoplankton. Now apparently we're supposed to call it microalgae, so I'm going to call it that. Depends on what field you're submitting your article to. So microalgae in the water column, Denali Ellis pictured here, which, by the way, Jim talked about algal blooms. I think those blooms are less likely to happen at this level of salinity, despite nutrients, because salinity does affect metabolism. But Denaliella are a little more flexible than some of the other algal species. So I'm going to do a deep dive on that, Jim, and get back to you on like exactly what we expect, because nitrogen uses at different salinities has been looked at in the lab with Denaliella. I just don't know it. So we know this, that there's sort of two food chains-ish, and there's crossover. And so these guys are thought to be the main source of food for brine shrimp. Obviously, brine shrimp also graze on the microbialite mats, but those of you who work with shrimp all day tell me that no, they really prefer the water column microalgae. Okay, next slide. And of course, the microbialite mats, which used to be thick and luscious and velvety, which are driven by a species called Euhalophyceae that I'll talk about in a minute, but have actually many microbes living in that mat environment. They become a lovely, nutritious bed on which the brine flies pupate. I'm not an animal scientist. That's why I'm bringing Dave Herbst here, because I don't really understand anything outside of a cell, and even whole cells scare me. I like the molecules inside the cells. So I'm not the right person to ask that question. But I have been studying the microbes that make these mats for about 15 years. Eric the senior and I had done some work with his graduate student Melody Lindsay that showed these euhalophysi. Go ahead. These cyanos. So this is my student Victoire in the summer, July of 2021. And you can see these luscious velvety mats. That's really the last time we were able to get samples of mats off these microbialites. This is on the south side of Ladyfinger Point, so in Bridger Bay. Okay, next slide. So this is from our paper with Melody, and this is the euhalophysi. Oh, you're not going to see my little animations because my arrow can't pop up now because it's not presenting mode. Oh well, never mind. Forgive me, I can point because the slides are low. So these euhalophysi species, this is the cyanobacteria the only cyano that shows up. And cyanobacteria are actually bacteria, and they look like chloroplast in plants or microalgae. So they're very, very tiny. And in evolution, they crawled inside a cell and made those bigger cells, made plants do photosynthesis. So they're like the organelle, the chloroplast, which actually has its own genome as well. So they're tiny. So we're growing these in our lab now, and that's part of the current funding that we have is to isolate and sequence the genome of these guys and study their resiliency to high salinity and desiccation in isolation so that we can get an idea what this species, take this off, what this species is doing that is driving this. So when we mush up a microbialite and sequence all the genes that are inside, we see these euhalophysi are a portion of the microbes. So they're not the only ones, but we know a cool thing that they do is they produce a sticky mat called an extra polymeric substance, EPS, and they help accrete all the calcium carbonate and hold it together. So they are the basis of the mat and one of the photosynthetic drivers. There are also some diatoms. Go ahead, next slide. And this is also pretty common in, say, the stromatolites of Shark Bay in Australia have a euhalophysi diatom combination. That bay is about 9% salinity or 90 grams per liter. So we did this cool study, Eric the senior and I, with Mike VanDevender, where the three of us were out at the lake looking at over 2 days various and I know Jim was there on at least one of the days, and maybe some others in this room were. But we went around the lake, different sites on Antelope Island, on the east side of Stansbury, and we found all these different sorts of microbialites that Mike had been observing. And he wanted to know, like, what's making them look different. And these are like these ridgy kinds, for example, around Buffalo Point, and there's some that look like mushrooms, and there's some that look like velvety mats that are super luscious, and they're a little bit different. And so we bet a 6-pack on, as you do in science, on the following. I said that the genes of all the microbes will all be different, and that's what's making them look different. It's the biology. And Mike VanDevender said, no, it's the wind and weather and where they're located and the physical parameters of the geology. And so Mike won, and this is the paper. This is the paper we published, and you can see, you don't even have to know what these species are. This is by order. But you can just see that they all the different sample sites, like the biology looks the same. And I put an arrow by the green is the euhalophysi. So it's a driver of all of these, even if there are these little ridges, the little carbonate ridges, the euhalophysi is driving the formation of those. Okay, go ahead.
Wait, where's Mike's analysis of the wind?
I didn't include that. It's in the paper. We've done this experiment, okay? We put a causeway in the lake. We made that part of the lake go to saturation. And this is what happens to the microbialites.
And we have also ground these up and done the work of trying to see who's there. And guess what? No Euheliaceae there, no Navicula, the diatom. These are just empty condos that Haloarchaea have moved into, essentially. So we've done that experiment. Okay, go ahead. So there are different structures, but similar consortia. The north arm structures are vestiges. We know that Euheliaceae is probably the primary driver of forming the mats, making the goo that sticks everything together, and that they're integral to the brine fly food chain. Okay, go ahead. So if we were to try to measure what we were trying to do, and leading up to the actual experiment, though, was funded by Forestry Fire State Land's Hot Topics grant, and this work was done 2020 to 2021. And if we were to measure primary productivity, could we infer the contribution of the microbialites? That was really the question we were trying to answer, comparing that to the phototrophs, the microalgae, and the water column. Okay, go ahead. So our experimental layout was both a spatial and a temporal approach. We did 4 seasonal samplings. One of the seasonal samplings, the controls weren't good, so I'm going to show you data from 3 seasons. We looked at 2 sites on Antelope Island, one that's rich with microbialites, and then an area where there weren't microbialites. We measured carbon assimilation with a radioactive assay. We measured chlorophyll. We measured microbial diversity, like looking at all of who's present in each sample. We looked at nutrients with Christine's group. She came along with us and sampled, and we did a nutrient analysis, which I won't show you today because it's a whole mega data. It's hard to show on a slide. And then we measured, of course, environmental parameters. And at the end, we tried to take all of that, use chlorophyll as a control, use our killed samples as a control, and model that in one big lake. And so I'll try to explain a little bit how we did that. Map versus water column. See, this is where my animation would circle this for you when I'm talking, which it's not working, so forgive me. So remember, what we're looking at are the algae in the water column versus the mats on the microbialites. Okay, go ahead. So we were at this north end of Antelope Island. This place near the marina where these microbialites are on the north side of Ladyfinger Point was north of Antelope Island, NAI, Site 1. Site 2 is White Rock Bay. And we were in this part of White Rock Bay where there are no microbialites. There are a bunch over here near White Rock. Okay, go ahead. I'm sorry.
Oh, we're troubleshooting.
Oh, they're troubleshooting. Thank you.
While they're at it, Bonnie, the radiocarbon, was it radiocarbon or stable isotope carbon?
It's the C14.
Really?
Yeah.
Okay. Any special issues with getting permission to?
We did it in vials.
Oh, in the lab, okay.
Yeah, and we did it on site, but in vials, injected it into vials.
Okay.
And then took it back to the lab to analyze.
Just a comment while we're waiting,
Yeah.
quite a bit of the diet of your grebes are brine fly larvae. So,
Awesome. That's good to know. Thank you.
I mean, we think of them as just feeding.
It's feeding on the shrimp. Yeah.
But you see huge levels of grebes over the
Yeah.
microbialite areas. And
One of the things that we've been talking about is trying to come up with a list of birds that feed on the different parts of the life cycle of the fly. And I haven't talked to some of the bird people in this room about that. I've talked to John Neal and to Marcel and Max, and I'd love to put together, especially as we're thinking more about the flies, I'd love to put together a list of who eats the larva, who eats the pupa, who eats the adults, who eats adults on the water like phalaropes, who eats the adults on the shore. I think that would be, they feed so many sectors of birds. I think that would be really interesting.
I would second that.
Yeah.
That would be super
Wouldn't that be
helpful.
interesting?
Yeah.
Yeah. So that comment is really helpful.
I
Also
did
thinking
an
about
analysis, I
crossover.
did an analysis of the diets of all the birds
Yeah,
from other people's published stuff.
so maybe you have what we want.
Well,
Wayne.
yeah, I can't remember if we broke it down by,
Yeah,
I think it may have got grouped by federal and
well,
then
that's,
stages.
that's something we'd really like to put together. So thank you. And thinking about Dave's visit. Okay, so go ahead. This next slide is the, oh, it is presented.
We did it.
Yeah, yay,
I
thank you,
knew we
thank
did.
you. Who's the genius in the room? You're the genius. So this is just a picture. This is an app that a lot of us use in the field that will give you GPS coordinates as well as a photograph, which is kind of nice, and it shows the direction you're pointing your camera, which is good, and the elevation. So this is White Rock Bay, the area where we were. So you can see there's no microbialites, and this is sort of the microbialite-rich bay that we were in. Okay, go ahead. So the assay kind of goes like this. We use a little vial, and we are looking at the incorporation of CO2 that is radio-labeled into the biomass. So it's basically doing the process of photosynthesis. It incorporates the carbon, and then we can measure that. And then we do these killed controls on site that then we have like a measurement of any background that we can subtract. Okay, go ahead. This is over the 3 seasons where the controls were good. And in summer, and this corresponded with our chlorophyll data as well, but this is looking at carbon assimilation. The numbers are low. If you could just advance one time.
Oh.
Pat Bagley's brine shrimp is going to show up. We know that primary production goes down in the summer because of grazing in the lake. And so here we're looking at 2 different planktonic sites, site 1 and site 2, and then these are the mats from site 1. Okay. So we're seeing less primary productivity in the summer. And then in the fall, yeah.
Those carbon, what are the units per square meter, per liter? It's always hard to compare planktonic production with, production on the bottom. So usually it has to be done per square meter or something like that, but you don't have the units.
This is done in a vial, so we did not extend that to square meters. This is not done in the lake. So we're just measuring millimoles of incorporation.
So how much microbial mat did you put in the vial? It would seem like.
I have all of that.
Okay.
That we can go over that, but it's all normalized. So each seasonal sampling, and then we had triplicates of everything, and every sample, every site, every season. So I can go through all the details with you, Wayne.
Okay.
And send you that analysis. So in the fall, notice the 2 sites were really different, and this is even when normalized and controlled, and the mats were a portion of the primary productivity we saw. So if you were to hit the next forward button, so this really argues for a spatial approach. You can't just take one sample, and also seasons, right? You can't just take one sample on one day. That's a photograph of what's happening at that moment. You really need to take sites from different places and over time to really make sense of something like this. And then notice winter, once we normalized for the controls and with chlorophyll, there's nothing there. And so we think that, so I've since read a lot about photosynthesis and temperature. And at the water temperatures we were measuring, photosynthesis completely slows down. So that's something that you don't see with chlorophyll, because you get really high chlorophyll measurements, but it's actually the photosynthesis isn't happening. So that was kind of new to me because I've not studied photosynthesis in the past. So the MAT contribution, when we finally calculated all the samples for both the planktonic sites, the MAT when we used both the planktonic sites to say what's going on in the planktonic compartment, and then we use the MAT data. In the summer, we get something between 36 and 50% of contribution of primary productivity from the MATs. And in the fall, we get something that is more rangy because of the data having higher error bars. Go ahead, I'll show you what we did over the whole lake. Oh, look, I drew a circle. I forgot. I do this when, for like my students, and it really helps. Anyway, so then I talked to Christine about like, what's the best volumetric calculation, and she actually looked at elevation during the time periods that we sampled, and thank you for that. That was really, really helpful. And then we used Baskin's volumetric measurements for the south arm, that paper, and plugged all that into a model. And Eric the Younger did that, so I didn't do the modeling myself. And so we measured carbon assimilation in MATS versus water and extrapolated that to the whole lake based on, at that time, the distribution of MATS that were underwater and the planktonic compartment volume. And we come up with something that basically the MATS are doing about a third of the photosynthesis of the lake. And I think that is probably a reasonable number. We tried this lots of different ways, normalizing for DNA for biomass, because we also collected DNA for sequencing. So we used that to normalize for biomass, and then we used chlorophyll to normalize for biomass. And whatever we did to normalize, we kept coming up with that 1/3 number, which I think is kind of interesting. Okay, go ahead. So this is the DNA results. This is over the 4 seasons, and this is looking at the MATS. Yeah, I couldn't see that. Yeah, the MATS. So if you look at the MATS, the green is where the order where Euhalophyces sits. And so you see they particularly are happy during the fall when they're underwater. Go ahead. And then you don't see Euhalophyces in the planktonic sites, right? So lots of other things. And so this is over all seasons. And I'm trying to remember, this is actually a Haloarchaea, which is starting to take off in the lake right now because the salinity is getting so high, but this was surprising at that time because it wasn't quite high enough yet. Okay, go ahead. And then again, the other planktonic sites. So this is site 1 and site 2, where you see no Euhalophyces. Okay, go ahead. And so we're currently isolating this Euhalophyces now, which involves growing it on plates to get colonies. So that's the purpose of solid agar, is to get like colonies in isolation that start from a single cell, so they're all genetically identical. And now we have selected colonies, and we are growing up separate ones, and then we'll do that process again to make sure that they're truly isolated, and then we will sequence the genome of the ones we have. We have isolated this one, and we have isolated a filamentous cyanobacteria, which doesn't show up in our genetics. So we're really curious about if that has, that one is coming from microbialites in our aquarium and not microbialites which were collected 10 years ago, and not microbialites in the field. So I'm not sure what to make of those yet, but we might sequence those as well. Okay, go ahead. All right, so just conclusions, go ahead. I think these are bulleted. The water column and the MAT differ compositionally. There's different microbes in there, and they differ functionally. Go ahead. So we should not expect one compartment to compensate for the other compartment. So if the microbialites go away, those microbes and the things they feed should be noted, that will have an impact on the lake. You cannot just double dip here. Okay, go ahead. Even if you're a filter feeder.
So the microbial mats primarily occur in these marginal zones, and those are very vulnerable, and everybody in this room knows this, but when I talked about this at an international meeting this summer, I had to say some things that people didn't know. Okay, go ahead. And then we're afraid that this could reduce primary productivity of the lake system to 33%. And I don't know how to back calculate, like, as the volume of the lake reduces, right? And as these microbialites disappear, this calculation is gonna look different. So I'm really glad that we have the opportunity, funded by the tech team, to look at this before this terrible shrinking. Go ahead. So I've just got some pictures now. This is a great picture of mics because you see the exact microbialites. If you hit the forward key, I think I've got it circled. You see the exact same microbialites in this picture and this picture, and that was in the same year, the difference between July and November. Okay, go ahead. That was a flight last October, so almost a year ago. And you just see these expanses of microbialites exposed. More pictures, dead microbialites. So that's what we're dealing with now. That's what it looks like out there. And I know Elliot, Andrew, you guys have been out there a lot. I know Jim, you're seeing stuff from the water. So you
We
guys,
see it on the shore too.
Yeah.
The bird surveys, yeah, we see the same thing.
so you can echo some of what I'm talking about for the sampling, but yeah, echo. And those are those two papers. If anybody wants the papers, I'd be happy to send them. And we're working on writing up this study. Eric the Younger is now in Seattle on a postdoc, so he and Libby and I are working on it. So acknowledgements. I don't know why the rest isn't coming up. Should say hot topics out there. Okay, is that separate bubble?
I don't know how to keep clicking.
Okay, yeah, so I'm very grateful for the funding that allowed us to pull these experts in and do this work. And go ahead, one more slide, just question slide, and that's the flight that we took over. It's a little blurry, but yeah, exposed microbialites. So we're a little concerned. And Lynn, you asked the question about salinity, and I want to answer that while I have this picture up here, because this is not the only threat to microbialites. Euhalophyceae in our lab is consistently dying at 17% at 170 grams per liter, and we're measuring 18.4 in the open water. So we have exceeded the tolerance range for the primary driver that forms these mats. That means the mats will start to die. That's what we would predict, right? And I can't find mats now. I cannot, I wade out, I go out on a paddleboard. The mats that are healthy are probably beyond where I can reach, and Jim will have to start snorkeling for me or something.
You'll have to go about 4 miles from where you were at Bridge.
Yeah.
To the west.
Yeah.
You might be able to wade down there.
I probably could.
Pretty shallow. 4 miles away from shore.
You gonna come get me if I get lost?
Got a radio.
Yeah, so the threat of higher salinity is the same as the threat of these things being beached. They will die. They are dying. And we are seeing the pupa that would sit on these velvety mats, just, they're totally bare of pupa. The mats are gone. The pupa are washed up on the shores by the billions, and not the pupal casings that you're used to seeing, right? But these glossy, like we're living pupa. And my understanding from talking with Dave Herbst is that the salinity, what we would expect to see is either the pupa hatch too early and the flies are reproductively incapable of doing their business, and so they can't produce eggs. So whatever we're seeing, you guys talked about like extra larvae in the water when you're collecting shrimp, for example. They could be hanging out in the larval phase because they can't pupate.
They could be incompletely pupating, the pupa could be dying, or we could be forming adults that are not capable of reproduction. So those are some of the hypotheses around what we're seeing in the field. Again, I only know the microbiology, so I'm trying to bring in people who can help us think through what's going on with the flies, but I'm deeply concerned. Yes.
Do you have an idea of whether they're resilient or like, so if they were to be submerged again?
Yes.
Salinity would decrease?
That's an excellent question. And a lot of people point to the beauty of tide pools, right? About how tide pools, the tide goes out and the barnacles all close up and then they blossom out. And that's not the way a saline system works. There is a lot of resiliency built in to life that has evolved to live at high salt because there are times of desiccation. The microbes that I study from the North Arm can live inside salt crystals for millions of years. Elliot can talk to you more about that. So I believe in resiliency. The mat itself forms a protective layer, but if the mat is disappearing and disassociating, it's not there. We know that if you leave the system at salt saturation for decades, the experiment we did in the North Arm, we've done that experiment. You can go too far, right? No amount of fresh water put in the North Arm will bring back those microbialites because the organisms aren't present, right? And I think that's, so why we're doing this in the lab to study how salty can you halophysi be, and if we dry it up, is it resilient? Can we bring it back? And when we sequence the genome, there are genes, there are genetic capacities known to help recover from dehydration, desiccation tolerance genes. So we can look for those. We can ask, do those exist in this species? So that's what we're doing from the lab approach. And I think we don't know. We really don't know. My guess would be, yes, they're resilient, but is that after a year? Is it after 6 months? Is it after 3 years? We don't know what time frame that is. So we can do lots of replicates in the lab relatively quickly. We can't do that experiment in the field relatively quickly. And it depends on Mother Nature, really.
Okay, Bonnie, we have one more question and then yes, we have, well, sorry. Oh, I envisioned Anne has been having her hand up back there.
I'm just trying to wrap my head around this 33%, because the mats are only in very small areas.
That's right.
And to have 33% of all the primary production of the lake,
they're amazing.
That's
They're amazing because think of it, they're very concentrated,
right,
compared to microalgae.
And but do they? So the distribution of primary production is going to be really limited.
Yes.
Right.
So, yes.
And they cover 25% of the lake or something. And most of them are still submerged.
Right. But again, it's going to be primary production is pretty spotty. Right.
Yeah,
it's not
correct.
And it's the same with the waterfall too.
Yep. Good.
Thank you very much, Bonnie. And please feel free to follow up with Bonnie afterwards, catch her on the fly with more questions. But Pace has been waiting so patiently, and he contacted us several years ago. Jamie, I don't know if you remember, like 5 years ago, maybe. I'm sure Pace will give us the background on pork nuggets, and this is not part of the tech team grant. This was something that Pace approached us about, and we said, pork nuggets? Okay, tell us more. And so here we are several years later. So Pace, please feel free to share your screen or we'll just focus on you now.
All right, thank you. Yeah, this has been going on since 2015, so 7 years now, although we completed the work in Quark nuggets at the Great Salt Lake in 2018. So this shows the breadth of science that can be done at the Great Salt Lake, and I appreciate the opportunity to give you basically a final report. on that. So I'm going to spend about 3 slides on background, so it'll become more intelligible. 3 slides on the outcome of the research that we did, and then the remaining 4 or so slides on the impact that it has had. And for those of you who don't care much about physics, I'm a private company, a for-profit company, and my ultimate goal is to help mitigate climate change. So I'll link all this research to climate change at the end. So magnetized quark nuggets, which I will call MQN, dark matter, is consistent with the standard model of physics. So let me tell you what that means. The standard model of physics is the theory latter half of the last century that is maybe one of the greatest achievements of science ever, that plus general relativity, in that we now understand the forces and fields and particles, and it makes up, for instance, the helium atom is shown here. It's made of electrons on the outside and protons and neutrons on the inside, but those protons and neutrons are made up of quarks. And the assembly of all the particles that make up the standard model is shown here in the middle. And in addition to the quarks that make up protons and neutrons, like a proton is 2 up and 1 down quark, and a neutron is 2 down and 1 up quark, there are 4 other kinds of quarks for more esoteric particles. The standard model of physics says the most stable of those of the ensemble is a up, down, and strange quark, an up, down, and strange quark, as shown in the lower left-hand corner. And that's what composes quark nuggets. So if you assemble these into large numbers, they should be the most stable particle possible in the standard model of physics. The reason that is important is at the beginning of time, shown on the right with the standard model of cosmology, after about 65 microseconds, the universe is cooled to about 100 million electron volts, and these up, down, and strange quarks will precipitate out of the energy field and form quark nuggets. And then that, we think, forms, we being the MQN collaboration, forms the dark matter that contributes to the dynamics of the universe for the last 13.7 billion years. Now, the result of our experiment at the Great Salt Lake helped us, really was a turning point in our research and helps us understand that process in considerable detail. So I'll go a bit into that. And our symbol of a quark nugget is shown on the lower right, and it's the internal magnetic field lines, because these quark nuggets are strongly magnetized, as are protons and neutrons. But since they can be big, those magnetic fields can be big. These MQNs impact in the Great Salt Lake would deposit a lot of energy, dynamite worth energy. So that internal magnetic field is like the Earth's magnetic field shown here, and the material flowing into it, like water in the Great Salt Lake, would flow around the magnetic field because it would become ionized by the impact dynamics and distorted. And so the size of the disturbance would be much larger than the physical size of quark nuggets. Quark nuggets have the density of the nucleus, and therefore 10 kilograms is about a micron in size. We can calculate very reliably what the energy deposition would be. It's a function of the uncertainties in the surface magnetic field B0 at the time we started the program. And depending on the mass shown on this graph, you can get megajoules per meter. I'd like to remind you, 2 megajoules per meter is like 1 stick of dynamite per meter of explosive energy. So there should be easily detectable. And indeed, when we started this work in 2015, we did all these theories. We were on the Great Salt Lake for 1 year up at the north end of the South Lake, monitoring the lake and monitoring impacts and doing experiments on the propagation of acoustic energy. And from that, we inferred that we could see impacts from the whole lake, making this a wonderful Great Salt Lake Observatory. We published this results in Nature Scientific Reports and proceeded with confidence to make 3 large floating platforms for a 3-platform sensor system, which we established as the Great Salt Lake Observatory in 2016 through 2018. So each platform survived a hostile environment with 12-foot waves at the Great Salt Lake and operate autonomously collecting hydrophone acoustic data. To calibrate this, we needed to emulate those impacts. So we emulated with 0.25 megajoules per meter explosive. That's equivalent to about 1 gram MQN at a distance. And so let me show you what that looks like. So we calibrated all over the Great Salt Lake with these very modest explosions, and we discovered something unexpected. Instead of a nicely propagating acoustic wave through the media, the complex geology of the Great Salt Lake and hydrology of the Great Salt Lake, with the density gradients in the water and the stromatolites below the surface and the rocks further down and the soft sediments in between, make an acoustic waveguide which allows 250 hertz frequencies to propagate faster than all the rest, as shown by this yellow at 3.1 kilometers distance. But it rapidly attenuates beyond that. So at 4 kilometers, you get this blue signal, and that effectively determines the size of the Great Salt Lake Observatory we can observe. So we located our 3 sensors system in the southwestern edge of the Great Salt Lakes, as shown by the red. And that red is the 50 square kilometer area that we can reliably monitor. The outcome of our experiments is we observed 0 3-sensor events for mass greater than 0.0001 kilogram in our 50 square kilometers during the final half year, 24-hour days that we could monitor it. Now, 50 kilometers is actually 50 times the second largest area for a high-energy physics experiment. That would be the Ice Cube Observatory at the South Pole. And this observation, this null result, was exceedingly significant. It meant that for the single mass model, which at that time was the only model that was considered for high-energy physics and dark matter, then the flux limit we found excludes 0.0001 kilograms, up to 0.1 kilograms. But that was inconsistent with some of the other data we'd collected in Ireland. So it motivated us to calculate the mass distribution of these quark nuggets, as I'll show on the next slide. But it also showed we needed a much larger area or something to collect quark nuggets for a much longer time. We published these results as magnetized quark nuggets in the atmosphere last year. And these null results were the turning point for our dark matter. I had resisted trying to do the calculation of quark nugget formation because you had to go back to almost the beginning of time, 65 microseconds after the beginning of time, where the universe was relatively small, as shown by the leftmost pepperoni pizza diagram, and the up, down, and strange quarks aggregated together into essentially single up, down, strange quark ensembles. But remember, they're highly magnetized, and so as they ran together, they would magnetically attract, and the standard model of physics says that is a ferromagnetic entity, which will also explain magnetar pulsars with large magnetic fields about 300 times that of neutron stars. And they would aggregate into a much more heterogeneous universe, as shown on the right. They would continue that process until until it reached a steady state where the universe has expanded enough so that they don't interact so often, and the mass has grown to the point that there are few of them to interact. This is our result of our simulation from 0 picoseconds after the 65 microsecond point, 100 picoseconds, a nanosecond, 10 nanoseconds, and so on. On the left is the logarithm to the base 10 of the quark nugget mass, and that distribution is the cumulative fraction of quark nuggets up to 100% on the right. So you see that even at a millisecond, you have grown about 27 orders of magnitude in the largest mass available. That was a very important result because it showed that the initial aggregation time of about 3 picoseconds was much shorter than the decay time of these up, down, and strange singlet particles called lambda naught particles of 200 picoseconds. So aggregation outran decay and gave a convincing to us explanation of why the standard model of cosmology can explain dark matter. It also gave a broad distribution up to greater than a million kilograms. Think of a World War II submarine compacted into about a 50 micron diameter particle. And that explains the Great Salt Lake results in other phenomena. For instance, you may see in the media about once a year that there's peculiar craters that form, which are non-meteorite impact craters, according to NASA. And they're roughly small. Meteors can't make a small crater on Earth because meteors small enough to make such small craters break up in the atmosphere. But there was like a 12-meter diameter crater in Nicaragua in 2014, a very small one at the Salty Brine Beach on July 4th in Rhode Island in 2015, an event in Tamil Nadu, India in 2016, in which a person was killed and 3 others went to the hospital. And interpreting those with quark nuggets shows a wide range from about 50 micrograms up to 50 milligrams up to 1,000 kilograms. Now, in addition, there was an event in 1985 that we were able to get access to thanks to the County Donegal Natural Resources Commission to excavate. It formed a 12-foot diameter crater at the top, as shown by this young lady in 1985. But our excavations, we found a granite boulder at 4.7 meters, which was shattered by the input. We know that because of the shape of the crater showed the direction. And then when we went down to the bedrock of granite at the bottom, it was chaos. The plates were thrown all over the place. For something to penetrate 4.7 meters in steel fractured granite requires a mass density of greater than 900,000 kilograms per cubic meter, giving strong evidence that this and other non-meteorite craters were formed by magnetized quark nuggets. We've done other excavations in Ireland, which also support that. And then it occurred to us about 6 months ago that although we had always assumed, and everyone else had assumed, that the gravitational force on a dense quark nugget was so large, it overcame the pressure of all rock and anything else. So the destiny of a quark nugget would be the center of the Earth after it impacted Earth. However, we found 6 months ago or so that if the quark nugget falls into magnetite or taconite, a more complex form of iron-bearing ore, then the magnetic attraction of the quark nugget to the ore exceeds the gravitational attraction to the center of the Earth. And therefore, magnetite deposits should be accumulating magnetized quark nuggets, since these were formed by biological processes 1.8 billion years ago. We tracked the magnetite as it was mined through the processing into rod mills and ball mills, and inferred that they are transferred to the liners, which wear out and are reprocessed in a furnace where they become new liners, and scanned the bottom of 3 of the furnaces in a pattern shown here, and found that our control gave a cumulative distribution versus the bumpiness, which is the gradient in the magnetic field in Tesla per meter, shown by the dotted line. But then a furnace that has been processing these liners for 9 years showed a remarkably different curve with much higher peak grads per meter and much higher mean bumpiness grads per meter. And 2 41-year-old furnaces with 2 scans on furnace number 2 showed, again, much higher density. Now, if you take the difference, if we've modeled all this with ComSol computer simulations, and we found that all of this data is well explained by the accumulation of 300 kilograms of magnetized quark nuggets per year per furnace. 300 kilograms of magnetized quark nuggets per year per furnace. That's actually enough to collect and do something with. Now, this whole story then is from the beginning of time, and with the standard models of physics and cosmology, through the null result of the Great Salt Lake, through the modeling of the aggregation of quark nuggets into larger and larger ensembles, up to our data says up to 10 to the 6 kilograms, with the magnetic stopping that is confirmed, the same physics that diverts the solar wind from the Earth, through the first principles collection in magnetized quark nuggets, through the processing, through the accumulation in craters, to the possibility of intersecting them before they get to the final destiny of that furnace, and collecting quark nuggets of commercially valuable size. That's about a kilogram quark nugget, or assembling them, because a kilogram quark nugget is sufficient to contain a burning thermonuclear plasma, and therefore to bring modular thermonuclear fusion for carbon-free energy production, which is the goal of our company and of the MQN collaboration. And hence you can see from this that the results we did at the Great Salt Lake was the turning point, because it forced us to do that calculation, which made the sense of all of our observations. And for that, we're very appreciative. So let me, even though this research was not supported by either of the Hot Topics funds, I want to thank you for supporting research in general and our research through your tacit support by issuing the licenses. Jamie Barnes at Utah DNR and David Shearer at the GSL Harbormaster Utah State Parks. The Great Salt Lake Observatory MQN Dark Matter team, the physics team is shown at the top, and that includes Tracy Nielsen, who did the acoustics work from Brigham Young University, and Bob Baskin, now at Gestalt Geoscience Research, LLC, who did depth of salinity versus depth measurements, and therefore the acoustic propagation in the water. The rest of the team you see. I also need to give a shout out to Dave Gazzone of Gonzo Boat Rentals, and Chad Wardell of Diver Down, and Jim Cross of Cross Marine, and Ryan and Mike Rondi of MRFX, all of whom did wonderful contributions to make this work possible. And you can also look at MQN collaboration for more information. All right, I think we have our, looks like running over time. My apologies. But we got somewhat of a late start. I'm going to unshare my screen now. And if you have any questions, I'd be happy to answer them.
Thank you so much, Pace. Any questions? You guys
I'll attempt one.
Go ahead.
Bill's the only person
So-
that really understood.
Yeah, I totally understood everything. Pace, could you say in plain words why that broad distribution of particles was necessary to, how is that consistent with the result you obtained on the Great Salt Lake?
Okay, so think of a constant flux of interstellar dark matter coming in as our solar system moves about the galactic center through the dark matter halo, it's called, which gives the mass that holds the galaxy together. So it's a constant flux, and that means kilograms per cubic meter coming in. The larger the chunk of, the larger the mass of the average piece of dark matter, the fewer events you observe. So the Great Salt Lake, even though it was 50 times larger than the next larger astroparticle observatory on Earth, it was still small by the dark matter standards. Dark matter is very rare. And so the null result at the Great Salt Lake under the single particle approach was inconsistent with the craters that we observed in Ireland. So something was amiss. And what was amiss was the single particle approach, which had been the only assumption of all dark matter research to that time. So it turned us away from that assumption to do a real calculation. And the real, if you can think of bazillions of these things interacting, the ones that combine first have a stronger magnetic field to combine more rapidly with others around it. So the biggest grow bigger, the very small that get left behind stay left behind. And that gives that very broad mass distribution. Once we had the mass distribution, we knew what to look for. And that explained the Great Salt Lake result, the impacts every year of non-meteorite impacts, the magnetar pulsars, and it then let us calculate what we should observe in the magnetite mining industry in Minnesota. And that dovetailed nicely then with what we observed on the bottom of the furnaces.
Wayne?
What can we expect when that 1 million kilogram quark hits the Great Salt Lake?
You don't want to be around. The one it did, 1 million kilogram did hit off the coast of Ireland, about 100 kilometers off the coast of Ireland, on August 6, 1868. It slowed down by going through 100 kilometers of the Earth's crust. It emerged at the side of a hill and was observed by the assistant county surveyor of County Donegal, who reported what he saw to the Royal Society. No one observed the impact, but it's like a small nuclear device going off underwater. You don't want to be there. But what he observed was a rotating ball of light that came down and lasted 20 minutes, during which time it moved 100 metric tons of peat in 4 distinct formations. He reported it accurately. We went and found all 4 as he reported, because peat grows very slowly, 3 centimeters a century in that part of the world. And it also, we carbon dated the structures and found it consistent with his observations. So yeah, that and the other data helped narrow that uncertain the surface magnetic field to 1.35 plus or minus 0.35 teratesla. Now, that's a big number, but it happens to be pretty close to the magnetic field of a proton or a neutron. It's just that when you have something much bigger, like a kilogram is like 10 to the 27th protons or neutrons. So you have a finite magnetic field that you can do something with. And the fact that this a kilogram quark nugget has a useful magnetic field 1 billion times the next largest magnetic field that humans can produce. And that's why it can contain, in principle, a burning plasma. Good question.
Pace, I thought of you in Quark Nuggets a couple of weeks ago when a meteorite exploded over Great Salt Lake. Did you hear about that? And did you?
I did not. A couple of weeks ago, I was in the Baltic without communications. So, no, that must have been exciting.
Yes, I felt it and heard it, and I'm sure most people in this room did. It was pretty remarkable. And there are pieces of it now around Great Salt Lake.
But not on the lake bed. Do not
Yes.
drive on the lake bed.
But you may not drive your vehicle out there to find them.
All right,
But yeah, take a look and see what you think about that.
will do.
Yeah. Thank you so much for your time and all of your work.
A pleasure. Absolutely. And I let me express my personal grief at the dropping water levels in the Great Salt Lake. It is terrible, and I'm so sorry. So I hope you and your team can find some way to keep the microbiota alive and return. I really enjoyed Bonnie's presentation, so keep up the good
Great. We're working on it. Thank you so much.
pleasure.
Okay.
Bye.
Bye. All right, we're a bit over time, so I just wanted to say our December meeting, we can look forward to a presentation from Ed Hamill. He did it was a tech team grant awarded to him last year. It's a toxicology report. We'll be talking about, it's escaping me now, just exactly what in the realm of toxicology, but that's all I wrote down. And then Brian Footen will be talking about his sea kayaking and videoing of his first trek around Great Salt Lake, and he's looking to do more of that. But there would be room for another presentation or two more if a tech team member has a grant that they would like to present on, and or if there are other issues. You can let me know about those. And you can just do that by email or after this meeting. And I would like to throw out, because I've been asked by the IT folks to get meeting dates on the calendar for next year. So I would like to propose that we will be meeting on February 14th, Valentine's Day. Will be a great day to hold a tech team meeting. May 9th, September 12th, and December 12th.
Say that again, I'm navigating there.
February 14th,
February 14th.
May 9th, September 12th, and December 12th. And
do we have to bring chocolate?
There will be treats on Valentine's Day and perhaps beyond.
Are you saying that new funding is coming to?
So yes, that's what we will be using some of our funding for, is snacks for meetings.
Well, I think we decided that if things get worse at the lake, we're just gonna pass around Jim Beam or something.
Well, yeah.
I know it's the state, but.
Treats and other items.
Yeah. There'll be box wine in the women's bathroom.
Okay.
Can you send that out, those dates?
I will send them out in the email. Yes, of course. My email group for this group is like 150 people long. And so if I send out calendar invites for that, I get 150 in my responses in my inbox, and it's a lot. So I will send it out in an email though with any follow-up items. Does anyone have any last minute shout outs, news?
Yes. October 6th, which is the Thursday, Friends of Great Salt Lake is having its fall fundraiser, 6 to 9 p.m., the Garden Place, and This Is the Place.
Wonderful.
Thank you.
Thank you, Lynn. All right, you guys, we'll see some of you tomorrow, and we'll see you in December, if not sooner.
Thank you.
Thank you.
Laura, sign us up for